Hello, everyone. My name is Harini Balasubramanian, and I'm a research fellow at Pine Biotech. Today, I will be presenting my project titled In Silico Comparative Transcriptomic Analysis of Endemic and Epidemic Kaposi Sarcoma. This project was mentored by Dr. Raghavendran Lakshminarayanan. Before I begin into the details about my project, I would like to tell you a little bit about myself. I have a bachelor's degree in industrial biotechnology from Anna University, India. My research interests predominantly lie in molecular biology. So some example areas are microRNA profile analysis, toxicogenomics, precision medicine, cancer biology, and methylation epigenetics. I aspire to pursue my master's degree in molecular and cellular biology. So what is Kaposi sarcoma? Kaposi sarcoma is an angioproliferative multicentric malignancy that is triggered by the infection of KSHV, or Kaposi Sarcoma-associated Herpesvirus. It has 4 distinct clinical subtypes. First, epidemic KS, which is an aggressive subtype that is commonly occurring in HIV seropositive individuals. Second, the classic KS, which is found in older men of Eastern European descent. Third, endemic KS, which is prevalent in certain areas of equatorial or sub-Saharan Africa. And lastly, transplant-related KS, which occurs when an immunosuppressed transplant patient is also infected with KSHV. So KSHV is an oncogenic human gamma herpes virus, which is also the etiological agent that is responsible for not only Kaposi sarcoma, but also strongly associated with B-cell proliferative disorders, including primary effusion lymphoma. The virion binds to the cell surface receptor, following which the viral DNA migrates into the nucleus. KSHV exists between a lytic and latent life cycle. Upon infection, KSHV establishes a long-term persistent infection in humans where it manipulates multiple host signaling pathways and also modulates the expression of cellular genes to thrive in the infected cells. In general, KSHV infection is essential but not adequate to induce KS disease progression. Despite antiretroviral treatment, the prognosis for KS is often poor, with high mortality due to the presentation of late cancer stage. Therefore, to identify more accurate detection methods and therapeutic targets against KS, a better understanding of the viral and cellular transcriptomes in the cancer are necessary. So this is the information about the data set that I obtained for my analysis. The data was downloaded from the NCBI GEO platform in raw read counts. The study involved a total of 51 samples, of which 3 samples of normal skin were taken from 3 healthy individuals and the rest from KS patients. Here, the samples from the diseased individuals were obtained in pairs. That is, the biopsies from lesional skin and uninvolved sites on the same individual were collected. Raw counts for each gene from the human and KSHV transcriptomes were provided by the authors. This is the methodology and the flowchart that I followed for my analysis. Firstly, I grouped the samples into endemic KS samples, epidemic, and normal skin. Secondly, exploratory analysis or principal component analysis here was conducted using the following parameters. PCA was conducted first for all the samples and then for endemic and epidemic separately. Next, differential expression analysis using the DSEC2 algorithm and the enrichment and GSEA modules were also performed to understand the upregulated and downregulated genes. And these were the parameters that were used for the analysis. And lastly, human gauge analysis for a better understanding of the pathways was also performed using the parameters as given on the flowchart. These are the results of the PCA pipeline. And this is the result for the differential expression and human gauge results. So for epidemic KS and endemic KS, dot plots for geo-enriched pathways, CNET plots also for geo-enriched pathways, volcano plots, they were all obtained. And for epidemic case alone, the arachidonic acid pathway and retinol metabolism were significantly downregulated. Similarly, for endemic KS, the human gauge results showed that the ribosome pathway was also significantly suppressed. Now, moving on to the biological interpretation part of the results that we've obtained from our pipelines. So a majorly upregulated gene in endemic KS was SOX11. So SOX11 is a neural transcription factor which is highly expressed in mesenchymal and neural progenitor cells. It is also considered an oncogene because of its ability to induce tumorigenesis by promoting cell survival. So the increased transcript level of this gene can be implicated in the progression of the cancer, and it can be an important biomarker of Kaposi sarcoma, provided that the detection assay is also coupled with certain viral genes. And in epidemic KS, the ORF6 viral gene was most upregulated, and this was restricted only to epidemic KS. It is a gene that is activated during early lytic replication phase in AIDS-related KS. So co-infection with HIV in AIDS-related KS accelerates and aggravates KS development. So it is possible that the high levels of the gene product were also a result of the indirect role of HIV in disease propagation. Next, let's move on to the results of the volcano plot for epidemic KS. So PPEF1 and PCDH17, along with CEMIP, were found to be massively upregulated. So PPEF1 is significantly correlated with retinal degeneration, and PCDH17 and CEMIP were found to have positive correlations with cell-to-cell connection and epithelial mesenchymal transition pathways. So the overexpression of these genes in epidemic case patients is indicative of sarcoma progression. Next, let's look at the volcano plot for endemic KS. So first, if we see the S100A1 gene, it plays an important role in inhibition of microtubule assembly. So its increased transcription level can be implicated in promoting cell proliferation and migration, like the PCSK6 gene, and thereby the metastasis of cancer tissues. Moving on to the pathways, the lipid metabolism and energy generation pathways were significantly downregulated in epidemic KS. The lack of regulation in lipid metabolism was observed in both subtypes, but the transcriptional imbalance was more pronounced in epidemic KS. And this can be seen in the arachidonic acid pathway, as I mentioned earlier. The arachidonic acid is a multifaceted PUFA molecule that confers cell permeability, to the cell membrane, and also is also involved in producing prostaglandins and leukotrienes. So ARA, or arachidonic acid oxidation, produces many other crucial molecules that participate in wound healing, energy balance, and response to pathogen invasion. So the suppression of this pathway in epidemic KS can be implicated in the reduced immune response and dysfunction of the central nervous system. Next, another downregulated pathway in epidemic KS was the retinol metabolism pathway. So retinoids are known to inhibit uncontrolled cell growth, and the suppression of this pathway substantiates the significant anti-tumor efficacy of the current use of retinoids in treating localized skin lesions. And lastly, the ribosomal proteins pathway was suppressed in the endemic case samples. So this may be connected to the recent fascinating discovery that KSHV induces production of the specific ribosomes that are preferential for viral lytic phase mRNAs. So it's possible that the virus manipulates the cell machinery to suppress the organically produced ribosomes in favor of the ribosomal proteins that aid in viral lytic protein production. In conclusion, 6 genes in endemic KS and epidemic KS were identified respectively to be differentially expressed in these disease conditions. While the incidence of endemic KS is decreasing in developed countries like the USA, it is still very prevalent in sub-Saharan Africa and the Mediterranean. So till date, the detection of LANA or the ORF1 gene product is the golden standard for KSHV diagnostics. In this study too, ORF73 gene, which encodes for LANA, was found to be well upregulated in both conditions with similar fold change values. But how far a LANA-based assay can characterize the extent of case progression is not clear. So from this study, two potential biomarkers have been identified, one ORF6 and two SOX11. ORF6 could be a potential biomarker for detecting co-infection in KSHV in HIV-positive individuals. And SOX11, since it's heavily involved in propagating lesions and triggering angiogenesis, makes it another prospective biomarker for KS. So in conclusion, more research on the physiological and genotypic presentation of Kaposi sarcoma is highly essential to discover more therapeutic targets that can increase not only the longevity, but also the quality of life in people living with HIV and in KS patients. These are the references that I used. Thank you so much.